And if somebody is from the Western world, good morning to them. So today, as Ogniva has already mentioned, I am a graduate student at IISc Bangalore, and today I'll be talking about active systems or active matter in general, which is a relatively new field in physics. Like, it has started over the last decade or so, and physicists are actively involved in understanding living and metabolizing matter which spontaneously moves. They want to incorporate statistical mechanics principle on these systems and include it in the condensed matter manifold. I will explain one such system, and I will explain why that I will go against the convention and give a counterintuitive mechanism to it. So give you a brief introduction, let me start with the outline, what I'll be talking about today. So I'll start with a quick revision of Brownian motion, and then I will tell you why that is not enough and you need activity in systems, giving a brief introduction to active matter in general. And then I will take the case of a typical active filament, which is a rod-like structure, and why do we care about it and why we are interested in understanding its mechanics. Now, obviously, before you start to understand the subject, you have to see what people have done before you, and you get ideas from them, but often you get ideas for not to do like them. And here in this talk, at least, I will try to show you one such example as how I write against the normal methodology people do follow. And then I will show you the convention in such systems, defy it as my title slide says, and at the end I will try to give a counterintuitive mechanism to explain the macroscopic behavior of active filaments. So let's start with Brownian motion. I think all of you have seen it at some time point, right? And read about it, maybe not, but let's start with the most common example. Milk, everything is a colloidal suspension. So here is a drop of ink which is getting an uniform composition in a fluid. And why is it so? Why it diffuses is because the ink is a colloidal dispersion of dye or pigment in a fluid. And the major characteristic property of colloidal dispersion is that the dye or the pigment molecules are micron sized. And that is why when you see this dye or pigment in a fluid under microscope, it looks like this. This is the Brownian motion under a microscope. And these active particles, these Brownian particles are moving nowhere on average, but you can see that it has a random trajectory. And obviously, no average displacement, we all know, because it's random. But when you see the mean square displacement, you get a linear in time dependence. Now, this is the characteristic signature of Brownian motion, and often it is not quite true for other systems as well. Because if you are an object moving at velocity v, and you calculate the mean square displacement, it is proportional to v square t square. And this is not the case in Brownian motion. So this is the most important characteristics, that the mean square displacement goes goes proportional to linear in time. And the slope gives you the diffusion coefficient, which is nothing but the ratio of the thermal fluctuating scales to the drag provided by the fluid. Now you can get all of this from a microscopic point of view. I will not obviously go to the details, but what I will tell you is that the fluid that it is in, it provides a mean drag to the system. In addition, if the colloidal particle is immersed in a fluid which has bath particles whose mass is very small and time scales much faster than that of the colloidal substance, it will impart random kicks to the object. And this, due to this random kicks which is fluctuating, this particle moves in the fluid, ultimately getting this kind of uniform distribution. And this occurs on a much larger time scale, like here to here, it takes almost 30 minutes. Now, this is all good. You can explain most of the things you encounter in your daily life at micron scales with Brownian motion. But the question is this enough? And first, I will give you an example that I will ask you to calculate something. So we all are human beings, we all eat food, right? And we all break it down to smaller particles like colloidal substance. So I give you an average adult body height, and I give you the diffusion equation in 3 dimensions. And I ask you this question: how much time it would take for the particles that you normally eat and you break down to colloidal length scales, and which has a diffusion coefficient of 10 to the power minus 9 meters square per second for head to bottom transport. That is, if you eat, how much time it would take to reach the bottom of your body. And I will wait for answers from the audience. Anyone? Like, just give me a random, not random, just give me a of the order answer. Like, this is the equation in 3D. I have given you the length scale and you know the D. I have given you D also. You just need to calculate this time.
It seems like a nanosecond. I mean, if I see 10 to the minus 9 square per second.
But it will go by D. So remember.
Oh, by D. Yeah, that's right.
So 10 to the power 8 secondish.
Yes, that's
yes. Right. Yeah. So it's approximately, if you convert it into time scales, it's about 10 years. Okay. So, okay. Let's see. Yeah, it's about 10 years. And this is not physically possible, right? If you take a medicine, suppose for a wound in your leg, and it takes 10 years for the medicine to diffuse to that region, you are dead by then. So diffusion is actually not workable for biological processes. And luckily in our body, we have active motors or active transport mechanisms to overcome this diffusive limit. Examples include there are active cilia or filament-like structures in our lungs, which help in clearing out mucus or debris out of our airways. And if you get a wound, as I have said, in your leg or somewhere, there are active processes which occurs at the scales of minutes or hours to close the wound. So all of these are not diffusive, they are active. And what do I mean actually by active? I will give in a second. But let us see that, is it only in the human body that we see active phenomena? Not really. We see it all around. And the most common example is that of a bird flock. You have all seen, if you have went out up to your roof, you have seen bird flocks, you have seen ants walking in a line. So they follow this coherent motion. And what is the reason for that? I will try to give you an answer, but let's see that, is it only in this macroscopic scales that you see active systems? Not really. You may not have seen this example before, but this is a bacterial swarm in which you can see this rod-shaped bacteria trying to align along their body lengths with each other. And the question is, why is it so different from normal Brownian systems, so as to say? Like, why a Brownian system can't do this and an active system can? Especially for a micron scale particle, that is the colloidal scale, we already have known, such as I'm taking the example of this bacteria itself, I have already shown you that the fluid exerts two forces, which one is the mean drag and one is the thermal random drive. But in addition here, perhaps by using some food, the bacteria propels itself through the fluid, and its internally generated drive along a particular direction is much larger than the drive provided by the thermal kicks. And this is an important property which makes this drive irrelevant. And a collection of such particles constitute active matter. And physicists have come up with a particular description or particular definitions to say what these active units should follow and how are they different. For example, each of these units, as I said, the bacterium consumes food. It is some form of chemical energy, right? Whenever you eat or drink something, it's some form of chemical energy. And they convert it into mechanical motion. So if each unit is doing it, it's a non-equilibrium process, as is well understood. But the important factor differentiates it from driven systems is that the energy level is happening at the level of each unit, like each bird, each bacterium, each ant. And what that when the energy input is such level, it breaks the symmetry locally, not globally like driven system. It's not like a three-dimensional matter, you are giving some forcing and the boundary and the fluid or the matter is shearing. It's not like that. So this is the important characteristic of active systems. And as a result, you see interesting emergent properties, like you have seen in all these cases. You see self-organization or coherent motion without where there is a leader who is saying, okay, follow me, I'm going in the front. This does not happen. Each active system is trying to align with its neighbor, and that's how this kind of coherent motions appear. I have already shown you an example of self-healing. Crystallization without attractive forces normally you would not see, but in active systems you do see them. And today I will be discussing more on the last example, which is sustained oscillations in absence of inertia. And before I move on to this particular topic, let me give a brief cheer to the Indian scientist who was actively involved in founding the field of active matter. He was Sriram Ramaswamy, and he is at IISc currently, and he has written beautiful review papers. As you can see by the dates, they are not very old. There are obviously other people involved in the system, but it's something to be proud of that an Indian scientist is actively involved in founding a field. And let's now move on to this system, which I said I will be talking about the rest of the talk, which is active filament. And biologically, it has a name. It's called celium. Don't worry about it. I'll discuss what it is. Just that a filament is something whose diameter is much, much less than the length of the system. And that's what the celium is biologically. So these are whip-like appendage used by cells and tissues to cause fluid motion at the micron scale. Example, human sperms. The filament that you show, which is undergoing spontaneous oscillations to move through the fluid, is the celium. Similarly, you will see unicellular organisms, which here it is attached to micropipette. There are two of them which are beating in phase, and that causes them to swim through the fluid. Okay, fine. So I have shown you examples of active celium, but as I told you that each unit should convert chemical energy to mechanical motion. Where is it happening actually in these systems? So it happens in their ultrastructure. It's a cross section of such a celium, and if you cut it through the cross section, you will see that there are tubes-like structures. Like these all tubes that you see are these two doublets, which are called microtubules, and they form a cross-linked structure. And in one microtubule of a doublet, you will see dynein motors. And these are the active energy converters in our system that we'll be talking. So what do? They consume ATP, which is the chemical energy in this case, and they attach to the nearby microtubule and slide them. Now, when they attach to the microtubule and walk on the microtubule, normally the microtubule is just shear apart, but because this whole structure is cross-linked by all these protein structures, the sliding is converted into bending, and that's how the active motors convert the sliding motion, like just the walking motion on microtubule, to this beautiful bending structure of the system. And is it only used by unicellular organisms to swim through the fluid? No. Cilia is the key to our survival. And one important is that cilia you see in unicellular organisms is similar to the cilia that you find in our lungs. Remember, I showed you a video where there are active filaments in our lungs. So the ultrastructure is same. And so if you understand someone from this whole range, you understand basically the behavior from unicellular organisms to multicellular mammals. So how do they actually are key to our survival? One example is obviously sperm. That is the reason why we exist. The next example is left-right symmetry breaking in early embryos. Now, you all know that the heart is to the left and liver is to the right, right? Now, this is the ACM, like electron micrograph of embryo. And you can see there is these filaments, which are called the celium, which protrudes from it at tilt. And they cause a unidirectional flow in the fluid by rotating in this manner. So whenever they're close to the substrate in which it is, the flow is very less. But whenever it is away from it, the flow is large. So in this way, just by rotating, it creates a unidirectional flow, and that is responsible for the placement of heart to the left and liver to the right in humans. Not just in this, let me now live show you what happens in our lungs. Is that you will have a dense carpet of celium. This is for a protozoa, but the similar structure is present in our lungs, where you will see a ciliary carpet. There are thousands of with a slight phase difference with each other. So this is the schematic which shows that this and this filament are in phase, but all of the middle are having slight phase difference with each other. And you can see the fluid is moving to the left in this case. And people have explicitly shown that when you have such behavior, which are called metachronal waves, that is long wavelength modulation in phase, you have unidirectional flows whose velocities are much larger than if they were all beating in phase. And that's why the same mechanism is used in our lungs to clear mucus. Now, all of these that I have shown you are examples of collective behavior of cilia. But even before that, there is a lot of debate at the level of a single cilium, like the level of mechanics of a single cilium, which is oscillating at a particular frequency and has a length scale of around, say, 10 micron. Now, what is a cilium mechanically is I'm not interested in the detailed internal structure. So as I have already told you, it's an active filament, so it will consume some chemical energy and dissipate the same through its motion. In this case, it is spontaneous oscillations. And the filaments, by sliding and bending, that should be balanced by a significant source of dissipation. Now, this is very important. Statement in this case, because
Hi, Debasmita ji. Are you there? So I think there is a network problem. So
okay, am I audible again?
Yes, yes, you are audible.
yes.
Yeah, okay, fine. So I don't know what happened in between, but let's I think how much did you guys listen?
So you can continue from this slide, actually,
Yeah,
the beginning of this slide. Yeah.
Okay, fine. So I'll start from the beginning of the slide. So what I was talking, that I have told you, or I have shown you several aspects of collective behavior of ciliary beating, but there is a lot of debate even at the level of a single cilium. And I will be talking about it for the rest of the talk. And a single cilium beats at a particular frequency stably with a stable amplitude and has a length of around 10 microns. So it's a micron scale system. And mechanically, because it's an active system, it continuously consumes chemical energy and dissipates the same through its mechanical motion, which is spontaneous oscillations in this case. And because it has a stable amplitude, it is required that the elastic stresses that are generated in the filament due to the active motors working on the microtubule, sliding and bending, whatever is happening, it should be balanced by a significant source of dissipation. Now, the question is, do we know what is the source of dissipation here? Convention says that it should be external fluid friction because it governs dynamics at micron scale, at the scales at which this filament beat. And the question is why? This is characterized by a dimensionless number, which you have studied most likely. It's a Reynolds number, but I will give you a snapshot of what's happening. Is that you have your Navier-Stokes equation, which is the Newton's equation for a viscous fluid. You have the advective term, which is this. You have the pressure term, and you have the fluid viscosity in here. So Reynolds number characterizes the ratio of the inertial term to the viscous term. And if I do a dimensional analysis of this and this term, I get rho VL by eta, where rho is the density of the fluid, eta is the viscosity, V and L are the characteristic velocity and length scales of the system. In my case, it's the cilium that I'm talking about. And if I put in the numbers, because I know my system, I will get a number which is much, much less than 1. Now, if it is much, much less than 1, it is already understandable that the inertial forces are much less than the viscous forces. And this is the low Reynolds number regime. And that's why external fluid friction governs the dynamics of micron scales, not only active systems, even in the colloidal systems. And the fluid equation that I had written, the Navier Stokes, the advective term is negligible, and what you are left with a linear Stokes equation. All of this is so far so good. Elastic stress should balance a source of dissipation. Convention says that it is external fluid, but there is more to this. The role of hydrodynamics is highly debated in the collective behavior of cilia. So what I mean by this is there are several of literatures proving and disproving that hydrodynamics is actually responsible for interaction between individual cilium when they are in metachronal waves or when they are synchronous, two of them or a multiple of them. Now, okay, fine. It's highly debated in each of the paper, proved and disproved hydrodynamics or not. But let's see what actually happens at the level of a single cilium. Like how much the hydrodynamics play a role in there. But as I said, we first see what people generally do with active systems or say active filaments, because obviously I'm not the first one to start this field. It is a very old field and people were studying active filaments, and the general approach to take up a live cilia, in this case of bull sperm, they've attached the head to a substrate. They see the bending motion of the filament, and what they do is write down the equation of motion for the filament. It's in the classical regime, so you include, because I have already told you there are elastic stresses in the filament due to the active motors, it's sliding and bending. And I have included this in a red term. You will understand why by the end of this talk, I'm pretty sure. They include only fluid dissipation. They know these two from, they know, they approximate that elastic should be something of Hooke's law, fluid dissipation something should be related to the velocity of the filament, and the thing that they don't know is active terms. And what do? They write constitutive models for this active drive, and then they solve for the waveform and optimize parameters in this model to get best fit to the experimental one. Like in this case, the blue dots are the experimental filament, like the beat, how is it changing with time, and the red is the theory to it. All of this is good, but you start from an equation of motion and at the end you match to the experimental one and match through a modeling. But what happens is when you go through this system, you often don't get a unique solution. Different models for the active drive can result in the same observed waveform, and this has been the case. And this I called a reverse approach, equation of motion and going back to the experiments. I said, why do that? Let's do the forward way. And as I said, I will ride against the tide. I will use experimental measurements rather than to start from an equation of motion to calculate and compare these two terms. As I said, I am interested in this fluid dissipation term. How much role does it play for the beating of a single cilium? So I need to compare it with the elastic terms, and then I will get, if I get an equation of motion out of it. And in this whole process, I will try to build an theoretical active filament model. So they are going, what the literature has been going is from equation of motion to experiments. I will be going from experiments to equation of motion. And that is what I meant by ride against the tide in my title slide. So let's see, where do I get my cilia from? Like I need something. I get from this organism called Chlamydomonas. It's a 10 micron cell body, and each cilia has a length of 11 micron. But what do I want? I want a cell-free cilium. I don't want the control of the live cell, which controls how will it beat and what is the energy given to the system. I want to switch off this biochemical regulation so that only I can study the interaction of the fluid of this filament with the fluid, and the boundary condition will also be simpler. So what do? I cut these filaments, not by scissors, obviously. These are suspensions of cells. I give them an anesthetic and they shed off their flagella, or the cilium in this case. And if you just see them in water, you will see Brownian motion of the cilium. These are isolated from the cell body. But if you give to the solution an external energy source, which erstwhile in the level of the organism was provided by the live cell itself, here it is not there, so no ATP, no energy source. I give an ATP and you will see that each of the filament will beat on the surface. And this is like I have cut up your hand, but I'm giving you energy to the hand and it is still doing what it should do. This is a very interesting feature in this organism. That's why it's a very well-known model organism to get active filaments. And it is free swimming on the surface. And if you clamp one end of it, it is more clear that why it bits similar to that of a single cilia. Now, okay, I have got my cilia. Now, what do I do with it? First, I see that each point along the cilium is undergoing periodic oscillations at a particular beat frequency and at a particular ATP concentration. Like I can ramp up my ATP concentration and the beat frequency will change. But what is the closest thing that you have already studied in your undergraduate courses? And I have kept this talk for the undergraduate audience mainly because I have tried to decrease the much detailed involved analysis of. So what is the analog system that you have already studied? There is some activity which is giving force to the system and the filament is bending. It is in a fluid which is giving viscous resistance. So the analog is that of a force damped harmonic oscillator, which you have all studied in your undergraduate courses, I see. So it's a mass in which I give a force, a time dependent force. It is attached to a spring and it is in a fluid. It can be liquid or gas, which is giving rise to this damping force. I have drawn the damper, but it can be just because of the fluid that it is in. Now, when I apply this force, this mass oscillates, and I can write down the equation of motion of the displacement of the mass from its equilibrium position. We all know how to write it, right? It's mx double dot plus bx dot plus kx equals to the applied force. So what are each of the terms? I have an elastic spring force, I have a viscous damping force due to the fluid, and I have the force at frequency omega. And we all know what its solution looks like. It's A cos omega t. The transient solution decays, and it is a steady state solution which force. And what I want you to next look closely is the form of the amplitude. So this is the amplitude of driven oscillations, which is finite at resonance. By resonance, I mean when the applied frequency is equal to the natural frequency of the system, which is omega naught. I have a finite amplitude, so far so good. But what would have happened if I had no damping in the system? If this term was switched off, bx dot. The bx dot is coming here as gamma. If this gamma is off, I can already see from this equation that my amplitude goes to infinity at resonance, and that's an undamped oscillator. But have you ever seen any oscillator going to amplitude infinity in your real life? The answer is no, right? It's physically impossible, and that's why you always need a significant source of friction so that you are staying at a fixed amplitude. And you remember I have already told you that the filament, that is our celium, beats with the fixed and finite amplitude. It does not break apart, and that's what I mean by it should break apart if it needs to have an amplitude which is infinity. So remember, I will come back to this again, and that's why friction is very important in micron scale systems. Now, what do I do with all this analog? Is that I try to measure the same forces that are in a force damped harmonic oscillator. I have an activity which is supplying the force, which is the oscillatory driving. I don't know much about that, so we'll talk about that later. But I can try to measure the elastic forces and the viscous damping forces in the cilia system, which is similar to that for force damped harmonic oscillator. Now, how do I do? You see that the elastic term is proportional to the displacement and the viscous term is proportional to the velocity. So can I do it from that? So what I do from cilia is that I simultaneously measure the waveform and flow field. And how do I do that? This is beating at a particular frequency. Now these black dots that are suspended in the fluid are passive. Remember, they are just to follow the fluid so that I can know what the flow field, that means the disturbance caused in the fluid due to the motion of the celium is called flow field. Now, fluid is transparent. If I were not giving these black traces, I would not see how the fluid is actually mapping. So for seeing the fluid velocity, that is for observing it, I put in the passive tracers, I track them. And when I track them, I already said, what can we get out this data? We can get the elastic term from the waveform and the fluid friction term from the flow field. And this is the waveform of the filament line, just extracted from the experiment. And this is the flow field measured by tracking these small black dots or small black beads in the solution. So when the filament, and remember this is an instantaneous flow field, I have given a timestamp in here. So that means when the filament is in this position and it goes delta t towards this position, after time delta t, it has a orientation which is something like this. This is the flow generated in the filament due to this. And insist, and you will understand why by do it. But as I said, I want to calculate elastic and fluid forces. First, let's look at the elastic. This movie beating in a plane terms in the filament, even in viscous, but that's later. So for example, the tangent angle can be described obviously by the the tangent can be described by the tangent angle. The filament center line is given by the integration of it. But even before I actually tell you what are the elastic terms in the filament, I have already told you it's a force-damped harmonic oscillator. So theta, which is characterizing the waveform, should satisfy an oscillatory solution, right? So does that do that? I have the experimental data, I can find it out. So plot theta along the filament length for 3 beat periods. We see that not only does it oscillate, it has something of a traveling wave pattern. And if we fit a traveling wave equation like this to the experiment, we find a perfect match. Now, this part is very familiar, what you see from normal force-damped harmonic oscillators, but this is the term which is extra. Don't worry about it, because the filament is beating asymmetrically, like when it is going in the forward direction and coming back, the waveform is not similar, and you have a time averaged shape of the beat, and this offset is characterizing that. We are not interested in this time average shape. Rather, what we are interested in the dynamic oscillatory beat of the filament about the mean shape. Okay, so far and so good. I have waveform characterized by tangent angle theta. It satisfied oscillatory solution and more a traveling wave nature. Now let's find out the elastic stresses in the filament. So again, a quick recap that the dynein motors cause the filament to shear, that is to slide with respect to one another, and by the kinematic constraints that were present to curve. And that's how you get the beautiful periodic bending motion. So what can be the simplest elastic contributions for a celium? First is that of a bending. Like it is characterized by the curvature, which actually tells you how the tangent angle changes along the filament length. And if you plot it color plot it on the filament, you can see the portion which is most curved has the highest curvature. And how do you write the elastic energy for bending? It is similar to that you write for the spring constant. You write F spring is proportional to the displacement. Similarly, the bending moment is proportional to the curvature. With the constant, you have a spring constant for the spring force. Here you have the bending rigidity of the filament. And the second contribution that is there for the shear part is characterized by U. Now, what is U? You can, because the filament has a fixed length, even though the dynein motors are shearing it, because the filament has a fixed length, you can relate the shear strain or the shear displacement caused by the dynein motors by the tangent angle. You subtract the tangent angle at a particular arc length minus that of the base, and this is what I call the shear angle. This is nothing but this term. So theta is still our main important dynamical variable. And this is a pictorial representation to show you actually that you have a positive shear, which is U, which is the difference between these two filaments. So the axonym you can model it as just two filaments, which are separated by this diameter A. And when the dyneins on the top filament exert a force on the lower one, there is a displacement, shear displacement happening. And because they're connected by these elastic links, it is bending. But when the shear displacement, you can just characterize, as I said, by the tangent angle. And you all know the spring force, it is proportional to the displacement. Here, similarly, the shear moment per unit length is proportional to the shear displacement times K is the spring constant in here and A is the filament diameter. Okay, fine. I have the elastic contributions for a sliding and a bending filament. Now let's move forward to the viscous part of the problem, which is the flow field. Now I had shown you this already in, I think 2 slides before. And this is when the filament is going in this direction, which is called the power stroke. And when the filament is coming back, it is called the recovery stroke. Now during that instant of the time, the flow field is opposite, as you can see. Now, what do I do with the experimental flow field? Do I really need it? The answer is yes, because if you want the external viscous drag unambiguously, you need this flow. Now, how? Now, we all know that any object moving through a viscous fluid experiences a drag and creates, and because it's in a fluid, it creates a flow velocity around it, which is V. Now, what is the simplest model for external viscous drag? You can do it from resistive force theory. Don't worry, it's not something fancy name. It's just what you have seen for that of a damped harmonic oscillator. It's gamma times the filament velocity. Gamma is a tensor here, don't go into the details, but the fluid force per unit length is proportional to R dot. This is the simplest model for drag, and using the simplest model for drag, I can theoretically compute the flow field as well by giving a line distribution of these forces along the filament length. Like if I have a line distribution of forces at each point directing towards this direction, which is FV, and I know the Green's function, which is the solution for delta function source. I know the flow velocity at any point in time. Now, how do I compute it theoretically? Do I know all of these terms? The answer is yes. How do I know? I know filament velocity from the measured waveform. And I know this drag coefficient from the literature. Sorry. And the Green's function also from the literature. It's well known that if a rod is moving very close to a substrate, the Green's function is given by Lorentz-Blake tensor. Now, if I compute the theoretical flow field, you can see the direction is very similar. And if you compare along these black dashed lines, you see a perfect, near perfect match, if not perfect. So what does that show you? It shows that the only assumption in my equation of flow is this FV, which is proportional to R dot. This is true for an active filament. It proves that my RFT, or the resistive force theory, is correct. And therefore, I have given you a first-hand direct experimental verification of the hydrodynamic drag force. Fine. Now I have got elastic, I have got viscous. This is the viscous part of the force. Now let's try to write the equation of motion for the filament. Now, again, I go back to the force damped harmonic oscillator because that's the common example you have seen. You have the inertial term, the spring term, the viscous term, and the applied force, which is a function of time. Now, in our case, I have already told you it's a low Reynolds filament, low Reynolds regime. So inertia is irrelevant. And the activity, that is the applied force, is provided by the dynein motors is MA, which we do not know about. But what we know is the elastic contribution, that to bending and shear. We know the dissipative due to the fluid from FV, and they combine in a way to give you the equation of motion, which looks like this. Don't go into how you actually come from here to here. It's a detailed derivation. I have purposefully kept it out so that it appears similar to that of a force damped harmonic oscillator. You have the two elastic terms in here. You have the fluid viscous term in here, and you have the activity term in here. And what I want to do is compare between them, compare the magnitude, because I know this theta from experiment. I know all of these material parameters from the literature. Now let's do it. But before we actually compare, what we do normally is we non-dimensionalize this equation so that we can compare in unit-free terms. Now, what do you mean by non-dimensionalization? You scale the arc length, that is S, with the characteristic length scale of the system and the time with the inverse of the characteristic frequency. All the material parameters, they are now subsided and you can't see them directly in the equation of motion. So I will now use this equation and directly compare my experimental results for each of these terms. So I write here again, these are the passive elastic terms. This is bending, this is shear, and this is my viscous fluid term. What I plot similar to theta, I plot this term as a function along the arc length for 3 beat periods. Don't look at the numbers for the time, it's scaled with the number, that's why it looks so high, but it's actually for 3 beat periods and across the filament length. Now this is for the bending elasticity term, this term. Next we plot that for shear elasticity and similarly for the viscous dissipation through the fluid. What I will ask to look at is not at how the graphs looks, but rather at the numbers of the color bar. And what you will see that the viscous dissipation through the fluid is 2 orders of magnitude lower than that of the elastic term. So what does that mean? I have no dissipation in this system. The only dissipative source was fluid and it is much less than elastic stresses. So my amplitude should have gone to infinity, but no, I have stable oscillations. So a quick recap is that an active filament can exhibit such oscillations only when the elastic stresses, that is this due to the active drive, are balanced by some significant source of dissipation. And as fluid dissipation cannot account for it, consistency demands that the internal stress in the celium, that is the filament itself, has a dissipative frictional component. Now, consistency demands that, but is it actually true that the celium is not an elastic filament, which people have used till now? They have only used celium as an elastic filament, and the fluid drag comes and the dissipation comes through only the fluid drag. So let's see if there is anything in the literature that proves that celium is a viscoelastic filament. And yes, there are some evidences which show that passive biofilaments often have substantial internal friction. Now, why? Because they are slow structural rearrangements within the filament interior or fluid flow through the pores. Now there are few literatures which propose, and I have taken out the easiest example so that you can relate. This is the displacement versus time curve for a passive celium in response to shear forces. So I have a needle, I have 2 needles placed on the passive celium, which is, remember, passive celium means the dynein motors are inactive. So I am applying shear forces on the response of that. Now you can see that it is not instantaneous. Immediately it does not go from 0 to whatever number, it's 10. So that means there is a friction in the system, right? If this response is like this and not instantaneous, it means that there is a friction. So the close-up term is that you have a shear friction, and this is the same paper which gave the term for, which gave the value for the spring constant of an axonym, but they've kind of missed this subtle detail that in the absence of shear friction, the response should have been instantaneous. So we have proved that celium is a viscoelastic filament. It can have internal friction, but can this balance the elastic stresses? So let's invoke the general internal dissipation terms that should be invoked in the equation by the filament itself. So as we have the bending elasticity, which was proportional to the curvature, if you remember, the bending friction should be proportional to the time derivative of the curvature. Similar again to that of a force damped harmonic oscillator. You have kx for the spring term, you have gamma x dot for the viscous term. Similarly, you have a time derivative in the curvature for bending friction. And similarly for the shear, you have a time derivative in the shear displacement. And if you remember our old equation of motion, we only had the elastic term, which is this one. These two are same, just non-dimensionalized. These two are same, non-dimensionalized, only the elastic terms, and the fluid term, which is Gn in here. And there was active, which we don't know about till now. And let's drop the negligible viscous term, which is due to the fluid, because we know that it is almost like it's 2 orders of magnitude less. And invoke only the internal dissipative terms in the equation of motion. And this is what we get. Passive friction in these 2 terms, no fluid term, only active term. Now let's see whether they can actually balance it. I have already shown you these 2 plots of bending elasticity and shear elasticity. And I have already told you that their color bar is the order of 1. And now let's plot these 2 parts, which are the frictional parts, internal friction parts. And you can see that even though this is like 1 order of magnitude less, this is comparable with this one. And this is actually of the same order. So what do we learn from all of this? Is that internal friction can actually counterbalance the elastic stresses due to the active drive. And as a result, instead of the old equation of motion, I propose this equation of motion for active filaments. And the story in all comes to one conclusion, that even if the filament is beating in a fluid, the mechanics of an active filament lies all inside. The fluid is almost doing nothing. Like if you take the same filament, ideally in vacuum, it should do the same. They need a lot of things like a lot of ions to beat, but say hypothetically, it should do that. Now, to summarize what I have shown you that to understand the mechanics of an active filament, you need to measure both waveform and the ambient fluid flow that is caused due to its motion for one-shot determination of both elastic and frictional components of the system. So whenever you want to describe any active filament, be it biological, be it synthetic, measure both and compare. Are they of comparable orders? And then move with the equation of motion. And what we have found out for Celium is that we have given a direct experimental evidence that the hydrodynamic drag provided by the fluid is negligible compared to the passive elastic stresses of the cilia that are due to the active drive, which was the convention till now. And we have proven and given a counterintuitive mechanism that internal friction is essential to have a self-consistent understanding of active filament motion at this scale. And this is a small pictorial approach of my result. Elastic stress is much higher, it's heavier than fluid friction, whereas it can only be balanced by internal friction. And if you want to go much details, much deep, this is our publication very recently on this topic. And at last, I would like to thank my co-workers. Prerna, she is my advisor, PhD advisor, and Ronojoy is a collaborator who helped us with the theoretical models and everything. There are funding from Welcome DBT India Alliance. Prerna is a fellow of that organization, obviously from IISc. And last but not the least, your lab mates are the reason why you can work so efficiently in your lab. So thank them. You must know this girl, she's from our institute only, like from Presidency only. And two more important persons who are involved remotely in this work is Sriram Ramaswamy and Aparna Bhaskaran. So Sriram is at IISc. He was my next door neighbor a few months back, and in lab, I mean in the department. And whenever I had problems, immediately I would go to him, and he would give me a nice explanation to it. And Aparna was visiting us in winter term, and she is his collaborator. He's my lab mate, and we kind of talked about the problem, and she gave us nice insights about it. Now, the last part of the thanks goes to the audience, and I thank you for listening so far, and I'm open to questions now.
Thank you, Debasmita. It was a wonderful talk and thoroughly informative. So we already have a couple of questions in the chat box. So
Okay.
Pritam Manna asks, what is mechanical efficiency of a cilium?
Okay, so you can actually, okay, let me read it. It is possible that the black dot somehow interferes with the unloaded motion of the cilia. I mean, okay, yeah, it's, so is this question or the question you asked is the one?
Okay, so he actually asks two questions. The first one is the about the mechanical efficiency, and the next one is this one.
So you can actually, okay, so you can actually compute the mechanical efficiency from how much energy it intakes, how much energy it outputs. So it's close to almost 1, because that's how it maintains the stable oscillations.
Okay, so I think that answers your question. So
yeah.
The second one is it possible that the black dots somehow interferes with the unloaded motion of the cilia?
Okay.
And
no.
Then he says that he means that black dots will collide with the filament and exert some other approximate periodic force. So is that significant?
No. So, okay, I'll go to the details of what that movie was. Let me escape my slide. And you are talking about this particular video, I think. Yeah, that's right. So the general trend is what black dots you normally take. You take them to be much less in weight, specifically the mass of that of the filament, so that it traces the fluid, not a load on the fluid. It's weightless very much, like it is of the lowest in size that you can go in the diffraction limited optical regime. So it's 200 nanometers in diameter, and it does not give you a loading on the object. And even when it collides with the filament, it is tracing the flow due to the filament. It's not providing any load to the filament, if that answers my questions. Prito, I think he asked.
Okay. So, Ognivada, you can go on.
Hi,
Yeah.
Debasmita.
Does that answer
Nice
my
talk.
question? Like, yeah. Okay, thank you.
Yeah, so I just have a small question. So all of your experimental results basically depend on the fact that you're able to replicate the energy injection from the living cell through some sort of an artificial forcing, right? So what I want to know is to what extent
Yeah, it's not actually.
are they like similar or something?
Okay, well,
So what exactly
on
are you
what
doing
extent
here?
are they similar?
Yes,
Is that
yes,
your question?
yes.
Yeah, okay.
Yes.
So first of all, the energy that I am giving is the same energy that the live cell gives to the filament. Just that it gives automatically, I am giving it externally. That's it. It's not an external energy source. And if you see the motion of this clamp celium, and that's why this is so beautiful, that its motion is similar to that if it were on a cell. So for example, okay, let's see this video. So this celium motion is similar to this one. And so even if you work on this isolated celium, because it replicates the motion of celium on live cells, on any substrate, like on this, on the lung airway, single one I'm talking about, it's a very good model system, and any results you draw from it can be applicable to them. The reason why we what happens that I have not gone into the details, when you have this system, it can support another kind of elastic stress within the object. There is something called a basal body connection, which gives another source of problem. And there is biological sensing happening, like this is a live organism, it can sense how I should move. So I have wanted to delete all that and only include fluid and the filament itself. And that's why we cut this off. And this is actually 2 years of my PhD problem. And so this is important, and this is absolutely applicable to all live organisms.
Okay, so just one thing then. So you're injecting ATP, that is fine,
Yeah.
but the way you're injecting ATP is different from a living cell, of course, right? Is there a way to actually replicate exactly what the living cell does,
Yes.
or is
So
if
what
that is
I'm
not
doing,
not important here?
What I'm doing is the energy that a living cell provides is the ATP, fine, but because this system is self-sufficient, as you can see, because I have taken it out from the organism, but it is still beating in the same way as if it were an organism, just by external addition of ATP. So what do? I have a suspension of this, which is undergoing Brownian motion, and I just drop load ATP in the suspension. It's a chemical to me. And as long as I have ATP in the system, this filaments beats. After 40 minutes, the ATP is gone and they stop beating. So it's very much replicable. Like it's actually
So
as if on the cell.
looking at the behavior, you're saying that they are similar, so it's fine.
Yes.
Right? Okay.
Yeah.
Okay.
Exactly.
Thanks.
So the macroscopic behavior is what you call, and people actually, biologists have seen. They have seen that whatever there is in the internal filament, whatever you get here is similar. Like even if you see ACM images of that of a isolated to that of on the cell, it's similar.
Okay, so over to Jim Bangali.
Okay.
Yeah, Debasmita, it was a very nice talk.
Thank you.
I had a question regarding the internal dissipation terms.
Okay.
Basically, I want to understand how you are coming to the conclusion that the internal resistance will ultimately complement the external force, right? The elastic stress part. Yeah.
Yes. Because you can see this slide, right? Yeah. You can see it, right? Yeah. Okay.
Yeah.
So your question is basically what I wanted to say is that how do I know that they are balancing is by comparing them. This term, which is kappa, which is the curvature, I know it from my filament motion, filament waveform. And this term, which is the coefficient, I know it from the literature. Now, using these two terms, the similarly way as I have plotted this term here, I can plot the similar term here. I can see that their magnitude is of the same order as this. And that's how I say that it can balance the stress. Whereas fluid could not do that. Its magnitude was much lower than this.
This magnitude that you're talking about, you are
Is
getting that from some...
of this term.
Okay.
From experiment as well as the literature value, which is consistent across diverse literature. So I need a coefficient, right? Like the spring constant. So, or the viscous coefficient of a force damped harmonic oscillator. This I get from the literature. It's very well known. This part I use it from the experiment, and then I plot it.
Okay. Okay. So you get from the experiment. That's
it's a purely experimental conclusion that internal friction is needed in such system.
Okay. Thank you.
Yeah.
Okay. So, ma'am, please go on.
Nice talk, Debasmita, and also very nice to actually, hear you after a long time.
Yeah, it was nice to be back here too.
Very nice. I see that your dad is also present, so very welcome.
Oh, you know that?
Yes, I know I know the name of your father. Yeah, I think that was your father. Very welcome to.
Yeah, that's my dad. Yeah.
Yes. Very welcome to Professor Debasmita Mondal. Now the question is that I did not understand one thing when you were showing the embryo and the heart is in the left and then the liver is in the right. Can you go to that slide and a little bit explain that?
Yes.
It's very interesting to see.
Yeah. So I don't have the movie, but what happens at the level of mechanics is that, let me show you again, that you can see this kind of celium, right? They are on the mouse embryo and they are tilted at an angle.
Yes.
Now, when they rotate, this is a schematic of the same thing. When they rotate, they disturb the fluid around it.
Yes.
Now, when they are rotating in this part, when it is much above the substrate, it is creating a flow in the direction from right to left, as you can see.
Okay.
From right, this is a pictorial approach where the tilt is such that when it is much above from the substrate, the flow that is creating is from right to left.
I see. Okay.
But when it is close to the substrate, the drag is so high that almost the flow is zero.
Yes.
So what happens effectively is that, okay, maybe I'll use the laser pointer. It's easier. So what actually happens is that this rotational motion of the tilted celium creates a leftward flow of the extraembryonic fluid that is present. Yes. And that's how your heart and liver placement happens in the embryo of the cell.
Oh, okay.
Because I have a unidirectional flow.
Oh, wow, wonderful. Very good. Thank you so much.
Yeah.
Okay, so Konadi, please go
on. Wonderful talk, Debasmita.
Yeah, she's much familiar with the work. Okay, thank you.
So can you please give some physical intuition to how you're calculating the internal stresses?
Yeah, the physical intuition means you want.
So in the
you want how I get this equation of motion?
No, when you calculate the internal stresses, like right in towards the end of the talk.
Okay. Here.
Okay, let me see.
Not here?
Yes, this one.
Okay.
So you calculate this bending. Yep.
Okay, so you want to talk me about how I get these terms physically?
Yeah.
Right?
Yeah.
Okay. So as I had told you that the dynein motors which are attached to the filament, they cause the filament to shear, right? When this motor is attached to the next one, they can attach and detach. So they attach, they walk, they slide the filaments, and because they are cross-linked, they will bend. So I have 2 degrees of freedom. So think of something, okay, let me help you with this. So think of something as, this is a 2 filament model. You have an upper filament and you have a lower filament, the whole celium. And you have dynein motors of one walking on the lower one. Okay? And you have a spring attached to it. Now, when you apply force on the top one, the spring is from straight, it goes to a bending position, right?
Right.
The spring is stretched. Now it has 2 components, one along the tangential and one along the normal. the tangential part of that extension will give you the shear strain or the shear displacement. And the normal part will give you the curvature. And that's how you get these two contributions for elasticity. Fine?
I see, yeah.
And when you go for friction term, as you always know that we go for the time derivatives of the same term. And similarly, we get the frictional terms.
Oh, now I see.
Okay.
That's wonderful. Thanks.
Yeah.
Okay, so I don't see any more questions. So Debasmita, I had actually one question and one comment.
Yeah,
so,
please.
So I'll first go with the comment. So I think a very recent study in August, actually, in this year, revealed that the sperms actually have fooled us for 350 years. They actually don't swim, but they spin.
Yeah.
So have you read about it? I mean,
yeah.
So actually what happened? I mean, can you incorporate it into this model and maybe improve it?
So a difference between sperm and the celium that I'm talking about is in the length scale, okay? So because you can see this filament motion is very planar. So the spinning motion cannot happen at this particular celium. So what happened in that paper, previously we all thought that sperm cilia and other cilia like that in our lungs and that, you know, have the same structure and also should function in a similar way, like this one, right?
Yes.
But you can see here it goes in a spinning motion towards the end, which you will not see because of its length. It has a very long wavelength. So that paper kind of distinguished sperm cell from other kind of celium and given a new way to look at it, which I am not sure how it will actually translate to to the celium, or this is 11 micron, whereas sperm has like 50 microns length of this filament. So I'm not sure how it will translate, but yeah, like I am also in thinking that what that should happen. But I'm sure that it is not fooling us with the spinning motion, because you can see it's almost beating in a plane. If it was spinning, you would see that towards the end something is happening.
Yes.
So that is, to answer your question in short, that is a futuristic goal of the study.
Yes, of course. So, and the question, so when you treat it as driven damped, so when you treat it as a driven damped oscillator, so we often see that the parameters used there are very, very sensitive. I mean, they can lead to chaotic behavior as well. So, I mean, is there any possibility that we can end up in having a chaotic motion by, tuning the parameters like that?
By tuning the parameters, meaning the elastic and the viscous parameters of the system?
Yes, and the F0 term as well, or
as
like
the dry. You know, this filament, even though I give it ATP, let me answer the second part of your question first. Even if it seems that I will give more ATP and it will turn into chaotic, it does not happen so. What happens is, beyond this 1 millimolar ATP, it stops beating. Okay. So till then, it is all, and beyond a particular ATP concentration, however energy you give, it does not do so. But to answer your question, this filament has a particular bending rigidity and particular spring constant that you can't change it like that. But obviously you can prepare synthetic filaments, right? You can, people actually prepare synthetic filaments for drug delivery and many other machines. Like micro machine is one of the newest new technology that people are trying to achieve by understanding this kind of filaments. So if you prepare such a system where you fudge with the B and the K, and you have such a system where you can apply as much forcing as you want, you can actually end up in getting chaotic motion. People have seen active turbulence for sure. So for this kind of filaments, I'm sure if you prepare something synthetic, you will get. For this, you can't, because beyond a particular energy, how much energy you give, it doesn't care. Rather, it cares by just stopping the beat.
So
it's a finicky system is all I would say.
Okay, so I think we don't have any more questions. So we have an interaction session here. So
Yeah.
After this talk, so I will request ma'am to please hand over, and just a request that before beginning the interaction session, I just want to share the screen and announce the next day's poster.